Buenas tardes, sean bienvenidos al Seminario Departamental que esta semana corresponde al Laboratorio de Biotecnología Ambiental. Y bueno, para hacer la presentación de nuestro ponente que en esta ocasión nos va a acompañar de manera virtual, tenemos a nuestra coordinadora del programa de maestría, la doctora Ninfa María Rosas García, quien va a hacer la reseña del ponente. Doctora, bienvenida.
Buenos días a todos. Gracias por estar aquí. Más bien la presentación que quiero hacer del doctor Temidayo es más bien en mi carácter de responsable del Laboratorio de Biotecnología Ambiental y quiero presentarles a este ponente. Pues el doctor Temidayo Oluyomi Elufisan es un investigador en biotecnología especializado en genómica microbiana, resistencia a los antimicrobianos, metabolismo lipídico y bioinformática. Obtuvo su doctorado en Ciencias en Biotecnología en el Instituto Politécnico Nacional y cuenta con una maestría en Microbiología Farmacéutica de la Universidad de Ibadan en Nigeria. El doctor Elufisan cuenta con una amplia experiencia en investigación en genómica bacteriana, incluyendo el aislamiento, la secuenciación genómica y el análisis funcional de patógenos ambientales y clínicos. Su trabajo postdoctoral en el USDA ARS en Beltsville, Maryland, se centra en la caracterización genómica de parásitos y patógenos transmitidos por alimentos. Mientras que en su investigación previa en la Universidad Nacional Autónoma de México, exploró la biosíntesis lipídica y su papel en la virulencia bacteriana. A lo largo de su carrera ha contribuido con más de 20 publicaciones arbitradas y presentado en congresos internacionales como ASM Microbe y FEMS. Es revisor activo de revistas líderes como Current Microbiology, Genes y Antibiotics. El doctor Elufisan se apasiona por aplicar las innovaciones biotecnológicas a soluciones sostenibles para la salud y el medio ambiente. Asimismo, es miembro de la Sociedad Americana de Microbiología y de la Sociedad Canadiense de Microbiología. En ese sentido, el doctor Temidayo ha estado colaborando con el Laboratorio de Biotecnología Ambiental, ha estado trabajando con nosotros, sobre todo en la parte bioinformática de algunas bacterias que tienen interés particular para el laboratorio. Como sabemos, uno de los objetivos desde siempre del laboratorio ha sido la búsqueda de bacterias entomopatógenas. Sin embargo, en esta búsqueda a veces encontramos microorganismos que, si bien pueden no tener un potencial entomopatogénico, sí pueden ofrecernos otras de sus características para poder ser utilizados para bien de la sociedad. Entonces, sin más preámbulo, yo los dejo con el doctor Temidayo para que escuchen la presentación. Gracias. Adelante, doctor Temidayo.
We'll be talking this afternoon on the topic title that says Whole Genome Sequence Analysis of Pigmented Bacillus inaquosorum LBA001, which revealed the biosynthetic gene cluster as for some secondary metabolites. Now, okay, so what do we want to talk about this? As I go on, I want to give a quick introduction of what it is. Bacillus inaquosorum are gram-positive bacteria, and they were once known as member of the Bacillus subtilis complex. It takes a long time before it could be differentiated. Many bacteria in the family of Bacillus inaquosorum has always been classified as Bacillus subtilis, but in 2019, some group of scientists did some deeper genomic analysis on them, and they realized even though phenotypically these Bacillus subtilis complex are very closely related, when they did some comparative genomic analysis, they were able to show that some basic characteristics differentiate them, and as a result, they classified B. inaquosorum, B. siebensis, B. valensis into their own species. So as one of those that they classified as B. inaquosorum, which was initially known as B. subtilis inaquosorum, but now it is now B. inaquosorum species. And why did they do this was because one of the major reasons was because they identified that B. inaquosorum produced a special kind of secondary metabolite that is different from every other member of the B. subtilis complex. So one of the other things that we note about them is that most of these B. inaquosorum produce pigments, and these pigments become intensive when they are subjected to stress. Other bacillus does this too, but their kind of pigment is not like other ones, and as a result, they group them based on this too. Now, apart from that, like every other bacillus, B. inaquosorum is spore forming, and sometimes they produce spores. So, B. inaquosorum can be found in different environments, like bacillus species. They've been isolated from barren soils. They've been isolated from agricultural soil, they've been isolated from marine, and also hot sulfur springs. Now, one thing about the B. subtilis complex is that they've been associated with entomopathogenic activities, of which B. inaquosorum is a member and could possibly be part of those that have that property. However, no studies right now has established the role of any B. inaquosorum as an entomopathogenic bacteria, but some beautiful characteristics have been reported about them, such as having antimicrobial capacity, like antifungal and antibacterial activity. One of such is Bacillus inaquosorum strain K27, K2R7, which has been shown to suppress Fusarium, a fungus that caused wheat rot of tomato plants. Now, B. inaquosorum, looking at them from the genomic perspective, they have a size of about 4.5 MB. Actually, not all of them are this size. Some are smaller or some are slightly bigger. And their TC content is between 4.35 and 44%. Now, they have a genome that is rich in many adaptive genes, and these genes have helped them survive in different environments. Like I said earlier, I mentioned that they could be found in barren soil, they could be found in agricultural soil, they could be found even in hot sulfur spring. They've been isolated from such an environment. So this could be associated with their genes that enhance adaptive characteristics. Apart from that, some of them have been reported to produce plasmid, and some have not. Now, what are interests? Now, the focus of this work was actually to get an entomopathogenic bacillus, and then as a result, knowing that bacillus are useful biotechnological agents, and that they can be applied in agriculture, they can be used for plant growth, promotion, and also they have antimicrobial property. So I think we want to see a novel versatile pathogenic bacterial strain, and as such, we decide to go for an isolation of this strain, which I think Dr. Anifa has mentioned about what the lab does. Now, in the course of that, if we get a novel strain or a new strain, we want to do biochemical characterization, we want to do molecular understanding of what it is, their biology, and what could be their potential application. So in the course of that, these methods were adopted for the isolation and for the identification of novel bacillus that could be entomopathogenic. So the soil samples were collected from Monterey Road, I think Monterey-Losa Expressway, and 1 gram of it was transferred in sterile, was serially diluted in sterile water, thermally chalked, and then at 65 degrees centigrade, cooled on ice, and this was inoculated on NB, and they were incubated at 30 degrees, and as such expecting to see Bacillus of potential benefit. Now, I want to mention something before I move forward. One of the reasons why the heat treatment was done was because we want to select a spore-forming bacteria. Okay, so when we did this, we are looking for a pigment-producing bacteria that can have important entomopathogenic function. So as a result of this, we found out that there's a particular one that's producing brown pigment, and then we want to understand it further. So because of this, biochemical characterization was carried out to determine what type of Bacillus is this. And some of the characteristics that were checked include the catalase, the carbohydrate, the fermentation, the as it is said in Badge's manner of determinative bacteriology. Now, the pigment production was also evaluated to know what sort of pigment is this, but in the scope of this study, I won't be talking about how that was done because it's another part of the study. Okay, so getting this pigment producing bacteria, the next thing that was done was to send it to transfer the colony into LB broth, for which it was used for DNA extraction. Now, the colony that has been grown on the LB broth was used for DNA extraction with the Wisa Promega kit according to manufacturer instruction. And once DNA were obtained, the DNA quality was checked with nanodrop before they were sent out for genomic sequencing. So the purified DNA were sequenced using the Illumina sequencer at the Laboratorio Nacional Lambda IPN in Centro de Biotecnología Inómica. So once the reads were obtained, the reads were quality checked with fast QC to see how good they were, and then vectors and adductors were trimmed with trim galore, and then the clean read were assembled with Unicycler. Unicycler is a software for genome assembly, that is for de novo genome assembly. Now, once the assembly was completed, the next thing we do was to check for genome completeness, and then to also check to see if there are no contamination in the genome. So we did that with CheckM and also with Busco. Now, after doing that, the next thing that was carried out was, because the obtained assembly did not give us a complete closed genome, so we adopted an online tool called Medusa to scaffold the genome into one. So after scaffolding the genome to one, then we start the other downstream analysis, which include genome annotation. So annotation was done in 3 ways, okay? But I'm going to be talking about 2 that we did in the lab. The first one was with Procar, and the other was with Rust. Now, we did Procar to be able to get a complete file of annotation, that is the FAA file, the FASTA file, the TFF file, and also the gene bank file. Then while we did Rust annotation to give us a quick overview of what is contained in the genome, so we could be able to talk about that. As I go in the presentation, we will see some of those things in my result. Then after annotating the genome, the next thing that happened was that we checked for some essential features in the bacteria. One of them is to find out, does it have resistant genes or not? Because it is an environmental bacteria, and because of the nature of the environment where they are found, it's possible to interact with different bacteria or different other organisms, and then we search for those genes for antimicrobial resistance using an online tool which is known as CAD. Okay, so after that, we also check for carbohydrate active enzymes, which are part of the enzymes that are essential for metabolic behavior in bacteria. So because of that, we search for that too with dbcam2. Then we also predict prosthetic cluster with anti-smash. These clusters tell us new functions that have not been seen or that have not been reported, or novel functions that have not been reported in bacteria. So we try to find that out using this. Then we also look for genomic island in the genome, because we want to understand, has there been any trait that we acquire via horizontal gene transfer? So this was done with Island Fiafio. It's also an online is an online software with which we did that. Okay, so the next thing we did after that is, in addition to all of those that I said, we also try to check, are there virulence gene? We did that with an online tool that checks virulence, which is called VFV Virulence Finder. We also check if there are some insertion elements in the genome to see if the genome has acquired some element or along the line that has helped its adaptation. Now, after checking for all of that, we also want to check the relationship between this particular strain and other B. inaquosorum that has been reported. So we did that by doing the average nucleotide identity to see how close it is to other genomes that are of the B. inaquosorum relationship. So we did that by using a tool that we call GSPCWS. It's also an online tool. Now, we also check again, in this case now we use the complete genome that we have to search for the 16S rRNA region. And this we do by subjecting that genome to an online tool called the eSetBio Cloud. Now, one thing I want to say is that this eSetBio Cloud not only gives us 16S, it also checks for contamination in the genome. So it's doing two things at the same time. So when we extracted that 16S rRNA, we used this tool to draw a phylogeny, and that phylogeny was just to be able to show how close is this genome to all that was using a 16S rRNA-based genes or rRNA-based DNA sequences. So after that, after doing all of this, because we know it's a pigment-producing bacteria, it actually produces a brown pigment, wanted to see what are the genes that are responsible for the pigment production. So we wrote an in-house script, and then we are able to extract some of those genes that are associated with pigment production in LPA001, which is the name given to the bacteria B-inaquosorum that was isolated in the lab. So after that, we want to look at what are the results we're able to find with all of the analysis that we got. Now, the next thing is that the first thing that we did, like I said before, is that we saw a pigment-producing bacteria, and then this pigment producer, based on the analysis that have been done, the biochemical characteristic standard that has been said of Bacillus subtilis, it actually fell into the group, into the characterization or identification of the characteristics that have been given for Bacillus subtilis. So, preliminary identification with biochemical characterization tells us is B. subtilis. That's the first thing we were able to note. Now, the next thing that happened was that we tried to look at the genomic characteristics. Having established that this is a B. subtilis, then we went further to sequence. Now, after sequencing, we were able to obtain about 1 million reads, and then the assembled genome gave us about 4.3, which is close to 4.3 MB, which is what is said of B. inaquosorum. I told us that between 4.3 and 4.4 MB. Now, and we were able to close the genome to 1 context, and then the total number of protein coding sequence, that is the number of genes that will give us particular protein, we found them to be about 4,149 genes. Okay, then it has one operon of 16S, 23S, and 5S rRNAs. There are 73 transfer RNAs, 20 repeat region, and total of 4,312 genes were predicted in LPA001. So we have this total number of genes. When we remove 60 RNAs and transfer RNAs and other RNAs, then we left with protein coding, which is 4149 that I said. Now we have 86 miscellaneous RNA, like I told us before. The genome size is 4.3 MB, and the GC content is 43.19. That is what is the genomic characteristics of this bacteria. Okay. So afterwards, then this is showing that we look at what's overview of this genome. What are the things it contains? That's why I told us when I said initially that we did annotation in two ways, RAST and PROCA. Now, our reason for RAS is just because RAS can just give a quick overview of what we have, and there we have, in this table, you could see that we have genes that associated with DNA metabolism, about 56 of them, virulence and defense, about 40 of them, genes associated with pigments, prosthetic and vitamin, there were about 145 of them, protein metabolism, cell wall, iron acquisition, among others. These are some of the genes that, okay, a quick overview of what it is before we start to depth further. And the other image you are seeing is just that the circular genome map and some basic annotation of LBA001. Okay, so the next thing is now to check, like I said, we check for the antimicrobial resistant gene that found that are present in this, and this table is just showing us some of the genes. I'll be explaining all of this very shortly. All the results I'll be explaining them very shortly. So these are the genes that we found there, and many of these genes are actually associated with glycopeptides. Some of them are associated with beta-lactam antibiotics, and some of them are also some of them are also disinfectants. I'll be talking more about this as I move forward. Now, for the CASI enzyme, in total in this genome, we have 357 of them, but for those that have been classified into various groups, I have, in this table, we have, for example, I have 27 that are associated with glycosyl hydroxylase, the glycosyl transferase 13, lyase 6, exterase 8, and then these are the carbohydrate binding modules. We have 16 of them, but in total for this genome, we have 367 genes that are associated with carbohydrate active enzymes. We'll be talking more about them as I go ahead. Now, we also predicted secondary metabolites, and in this genome, there's some important, there are new secondary metabolites that were found in them. And one of them is the gene that was reported in this is different from the one that have been reported in other bacteria. And if you check their identity that you have, you see that it's kind of very low. It shows that it's not so close to other ones. That is sulfatine, pilipastine. They are kind of different from other being reported in other bacteria. And that's why the identity similarity is very low, as you can see on the table. Now, the next thing that we have, the next result that we have is it's the genomic island. Now, in this bacteria, there were 11 genomic islands found, and like I told us, we have 14 secretion systems, all of which are type 4 secretion system. We never find any secretion system belonging to type 2 or type 1, and this is typical of gram-positive bacteria, so it's not surprising. Now, we did not find a major virulent bacteria. This is a plus for this bacteria because if it's virulent, then it might not be useful for any biotechnological application because of its virulent nature. Okay, so then we did comparative genomics. Now, the comparative genomic analysis show us that Bacillus LBA001 is very close to other inaquosorum species, ranging closely to the family of Bacillus inaquosorum strain A65.1, BSC2012, and M3 is close there. It's come as an outgroup from that family. And one thing that I can show us from this tree is that this group of bacteria seems to be very diverse. I will be talking more about that as I continue. This is a kind of 16S phylogeny. Then this is another phylogeny that we did. I didn't remember to mention that in the method. This is called base phylogeny. In this one, we make of 106. That's all of the genome of Bacillus inaquosorum that were available on NCBI at the time of this study were compared and were analyzed. And then we use the core genome, that is the core content of all the Bacillus inaquosorum. We use them to draw a phylogeny to see how close they are. I'll be talking more about this too very soon. Now, we saw that LBA001 is very, very close to, even though it's forming as an outgroup, but it's close to a strain called NB9822 when we compare their core genome. That must be the closest strain to it, and it is supposed to have similar characteristics. Okay, now, the next thing we did, like I said, is to find the average nucleotide identity. And what is that? We try to check how close they are to other genomes. And from this, you will see that most of them shared 99.8% and above. And that's why those are the colors that you saw in red. They are between this color that I find shows that they are above 99. Many of them, even though a few of them were kind of different from, slightly different from others, but majority of them are close. And B inaquosorum share a lot of similarity with some of them. Okay, some of them, at least around 98%. You will see that there is no one that is less than 97.5% to show you that this is a family that is really very close, even though there's some diversity among them, which some of the reasons which I'll be explaining soon. Now, another thing, this is showing that we did a pan-genome analysis, which gave us that core genome that I show us, core genome finding that I show us, and the pan-genome shows that the pan-genome is still open, as you see that this guy is still increasing, and this is the pan-genome, this guy is the pan-genome line, and show that it's still increasing, and then it means, which shows that many more genome can still be added to this family or to this species of Bacillus inaquosorum. Okay, so the next thing that we had was to be able now to talk. I want to talk about what are the importance of what we saw. Now, the first I said, we isolated a brown pigment producing Bacillus from soil using a Tema chalk and culture technique. And our initial biochemical tests show us that it's a member of the Bacillus subtilis complex, and then also our genome sequence and ANS confirmed that it is actually inaquosorum. So one thing I want to say here is that it is an established thing that we should not rely on phenotypic characteristics alone to establish the nomenclature or the taxonomy of a bacteria. That's the first thing I want us to note in this place. Now, when we do the ANI, it shows that this strain belongs to Bacillus inaquosorum. So if we are going to be working primarily on phenotypic characteristics, we may be attributing some traits that are not. So that's why genome sequencing or genomic annotation identification is very molecular identification is very important when we're looking at bacteria. Then the other one is the CCNS also confirmed that this strain is B inaquosorum. So I won't be going further on this because I already explained now that molecular identification is very important when you are working with bacteria. Okay, the next thing I want to talk about is the size. I already said it. The size is between 4.2, is around 4.2 MB, which fall within the known for Bacillus inaquosorum, and the GC content is 4.31. We saw that the genome completeness, like I told us, was 99.1%, which showed that it is possible that it's possible that some fragment has been lost in the course of assembly or in the course of sequencing. So that's why we don't have a whole complete genome. So I showed us on that table that the genome has 4,149 coding sequences that translated into protein, 73 transfer RNA, and 20 repeat regions. We found genes that are treated with carbohydrate metabolism, amino acid biosynthesis, stress responses, antimicrobial resistance, membrane transport, motility, and sulfur metabolism. We found all of this, like as I show us in the table of the RAST annotation. Sorry, in the figure of RAST annotation. Now, sorry. Sorry. Just give me a minute. Okay. So now, from the pan-genome analysis, for 106 B inaquosorum strain, we found out that the pan-genome is still open. I've told us that, even though at the end of the day, they share only 189 genes. Now, one thing that this implies for us is that this strain or this group of bacteria have highly adaptive mechanism that differs from others. That's one thing that this shows us, that many of them, they have high number of ancestry genes, which are genes that talks about adaptation. That's why the core genome content is kind of small. What they share with their family is small, because I think their characteristics has to do with the environment where you find them. So that's why this is it. As a low core gene diversity indicates also that they have high lateral gene transfer, and then it is possible that evolution is going on along them. And this also might explain why it takes a long time before the Bacillus subtilis complex was separated. It has to do with that evolution was taking place over time, and as a result, this group of strains are adapting new features that make them adapted to the environment. So that is what we saw, what that implies. Now, from the ANI analysis, the closest strain to LBA001 is the strain EC3005 3005N E3 that shows that is the closest to it. Now, there's slight, like I said, there's slight genomic variation in LBA001 compared to others, and this is associated with its adaptive features. Now, we found out that there are 357 carbohydrate active enzymes in LBA001. They are glycosid hydroxylase, transferase, polysaccharide, esterase, and CBN. These enzymes have been reported to be associated with chitinase and chitosinase, which are essential for environmental adaptation, they're essential for biomass degradation, and also some of them have antifungal activity. Now, this activity also suggests that these enzymes that we mentioned have been reported to associate with ruminant digestion, bioremediation, and then implying that LPA001 can be applied in the agricultural sector. Now, our anti-smudge analysis identified gene clusters of secondary metabolites that include fedicin, sulfatin, bacillicin, subtilocin, and pulmonary bacillibactin, sulfatamicin, and bacillin. These compounds have antimicrobial, antifungal, and biocontrol property. The presence of of fedicin and sulfatin in the genome shows that this bacteria have antifungal and control activity, implies that it can be used in the control of plant disease and also in the development of antimicrobials. Now, one thing that could be scary is the fact that there are 13 genes that are associated with antimicrobial resistance, and these genes are efflux pump associated genes. They are beta-lactamase genes that are involved in altering or converting resistance to vancomycin. We have those that are converted to cephalosporins, carbapenem, macrolide phosphomycin, and aminoglycoside. Now, I want to pause a bit. Okay, there's something that it's important to predict antimicrobial resistance with molecular technique, but it's also important for us to phenotypically confirm them. Now, why did I say this? Sometimes some of these characteristics may not be manifested phenotypically, even though they have those genes. They may be reserved genes for what is happening in the environment. That's number one. The other thing I want to say is that it's also possible that they have acquired this gene because of the need to adapt or to survive in their environment. So it's possible that they have these genes just as a result to help themselves, or they have acquired it because of what is found there. So for us to be able to say this organism, they are a threat or they are safe, we need to do a phenotypic characterization of the antimicrobial resistant property. Okay, that's one thing I want us to take note of that in that perspective. Now, the next thing that we say is that when we do the virulence factor analysis using the virulence finder, like I mentioned before, the only thing we find to be associated with virulence is bacillibactin, and bacillibactin has been reported to be an antimicrobial agent. So it's not like it's a virulent bacteria, so it is safe to be applied as a biotechnological tool. Like now that's why I wrote that everyone is likely non-pathogenic and safe for biotechnological use. Now, we found 11 genomic iceland, which is about 8.4 kilobase, please, sorry, not MB inside. And this genomic island shows that this bacteria is participating in horizontal gene transfer, and it's not surprising. It's possible because they live in an environment where they need to survive. They may acquire new genes from related bacteria or even from other bacteria interacting with for them to survive. So it's not surprising they have some genes that are in the genomic island. Now, we also identified that there are 14 type 4 secretion systems, and then we do not have any type 2, we don't have any type 6, and which is common, like I said before, to gram-positive. And this type 4 secretion system, actually, many of them, they are also for defense and also sometimes they are for adaptation. So it's possible that these are all, they have acquired some of these secretion systems for their own mechanism of defense, where they acquire material or they expel material from their system. Now, we also have that this system is environmental regulation and defense against. Okay, I already mentioned that. Now, 4 insertion elements were found in this genome, and deeper analysis of them showed that it's only contributing to the genome plasticity and gene transfer. So, based on phylogenetic analysis of the core genome and 16S, we know that LPS is a distinct strain of Bacillus inaquosorum, and that it is really, really close to DE11, HT5.1, M3, and BS2102. And our comparative genome analysis shows that this genome is still undergoing evolution and is actively acquiring gene via horizontal gene transfer. In secondary metabolite. Now, I'm looking at what is the potential use of this bacteria. It's rich in antimicrobial secondary metabolites, which suggests that they can be good tool for bowel control. And also because many of these secondary metabolites I mentioned, like I said, like pegicin, like surfactant, like sotilocin, they have been reported to be antibacterial. We can adopt them for development of new pharmaceuticals. And also, we found that the case enzymes are rich in it, and then some of them have been involved in the control of plant pathogen as previously reported. So because of that, we think high number of case enzymes shows that this bacteria can be applied for plant pathogen suppression, biomass degradation, and then can be employed in ruminant feed to help in digestion of chitin, as we found chitinase in it. The genomic trace supports their use as a possible probiotic or bioremediation agent and valuable producer of biomolecules. Now, we also found that they be useful for environmental purpose because of their antimicrobial property and also because of the production of secondary metabolites. LPO1 seems to be genetically diverse and have shown good environmental adaptation, and then it's a promising biotechnological tool. Its genome has so many numerous secondary metabolites, resistant genes, which are valuable resources for antimicrobial production and environmental application. So far from this study, we have seen that LBA01 is safe, and because it does not have any significant virulence factor, showing that it can be applied in the industry with relation to agriculture, pharmaceutics, and bioremediation. At this junction, I want to give thanks to Dr. Anifa for giving me the privilege to present this article, which is presently undergoing a kind of review process, and also to give this work in this seminar. Thank you to everyone for listening, and thank you, CBG, for giving me the opportunity to present in this seminar. Thank you.
Ok, agradecemos la participación del doctor Temidayo y damos paso a la sección de preguntas. Si alguien tiene alguna, aquí están los micrófonos.
Gracias. ¿Hacia dónde me dirijo? Okay. Do you have in mind following this study by doing in vitro or in vivo assays like in pharmaceutics or in another field?
Oh, okay. Thank you for the question. Can we hear me, please?
Yes.
Hello? ¿Me escuchas?
Sí.
Okay, thank you. So thank you for the question. You see, for the scope of this work, my aspect is to look at the genomic overview of the genome, the importance like I've shown. Now, it is left for the lab I'm collaborating with to continue to see the in vivo. The only thing I've been able to establish is to show that, okay, this is a potential beneficial bacteria based on the concept of the genome, but it may not be within my purview to be able to talk about going further into the step. I think the best person that will answer that question is Dr. Anifa, because she's the effort of the work. So she determines the next thing we have to do. So, but for now, what I'm able to show is that this is a potential bacteria for antimicrobial use or biotechnological application. Thank you very much.
Thank you.
Good afternoon, Dr. Dayo. Thank you for the presentation. My own question revolves around the secondary metabolites. How are they synthesized? Are they synthesized by ribosome or they are non-ribosome synthesized?
Okay. You see, the secondary metabolite that were predicted, they are of two types. Some are synthesized by ribosome, and we have some that are not synthesized by ribosome. Let me go back to the table so I can show you some of them. Just give me a moment, please. Oh. I don't know, maybe I have the table, the identification, the explanation in the table. Okay, thank you. One moment, please. Okay, now, you see, all of these ones that they wrote NRP, they are the non-ribosomal synthesized ones. All of these NRP. But like these ones, like the one that they write ORDA and CDPS, Santipeptide, they are ribosomal synthesized. But the ones that you find, you see that they are NRPs, they are non-ribosomal. So it's from both ends. So we found those that are ribosomal synthesized, and those that are non-ribosomal synthesized. So that's it.
What do you consider is the less difficult? What do you think? What do you think is the less difficult method to assemble or annotate these genomes you work?
Okay, the simplest way to annotate the genome.
Yeah, because at this moment I'm trying to do the same that you did in your work, so I didn't choose what is the less difficult that I can use.
Okay. Now, the first I want to understand is that when you're working with genome, it depends on the genome. As I'm not going to tell you this is the best, this is the less difficult, this is the most difficult. But one thing I'm going to advise the best is this. Now, if you're assembling a genome, first let me start from the sequencing method. Now, because sequencing are kind of cheap, I think it would be good for you to use two sequencing methods to be able to make it easy for you to assemble them. First, you use the long read and the short read sequencing. That is like Illumina and then like the PacBar or Oxford Nanopore. But if because of financial resources you can't afford the two, now if you are going with one of them, I would advise basically the PAM, that is the second generation, the Illumina family, because the long read will give you good reads, but there could be a lot of repeats that may affect your assembly. But if you are using the Illumina, I would also advise that to make things easy for you, you can do a reference guided assembly. Is that clear, please? Hello?
Yeah.
Okay. So if you use a reference guided assembly, it will help you to close the genome very easily. But if there's a problem, another thing I would advise is you can compare different sequencing methods, different assembly methods. For example, but presently now from my experience, I'm going to tell you Unicycler is working effectively well. So if you work with Unicycler for prokaryotic genomes, it's very good. It's very, very good. Work with Unicycler, and if you have some limitation, then we can talk about it. You can always get my contact and you may share with me if you want me to be of help. For genome assembly, I think to some extent I have some strength and we can always work together. But Unicycler is very, very effective now, but it's giving some limitation. There are some other things I can help you, I can talk to you about, which you can use and to help the assembly and to make the work very easy and very fast. Assembly is very, very easy to do now. It's no longer complex because there are so many tools that you can use now and that will make the work come out very fast and very easy.
Thank you.
Agradecemos la participación del doctor Temidayo y en estos momentos proyectamos el reconocimiento que le vamos a hacer llegar a su correo. Agradecemos también al Laboratorio de Biotecnología Ambiental y a todos ustedes por su asistencia. Muchas gracias y buenas tardes.
Thank you.